Right then, shall we? Okay. Good morning, everyone. It's a pleasure for me today to introduce to you Professor Rodolphe Barrangou. So I'm sure many of you already know him. He rose to prominence when he and Philippe Horvath, while working on bacterial immunity, landed upon CRISPR. So in a 2007 paper, they showed that these clustered regularly interspaced repeats, which nobody had really paid any attention to prior to that, were actually a bacterial adaptive immune system. And in one paper, they demonstrated that these repeats and the spacers between them are associated with gain of resistance to viruses, that the gain of such a repeat and spacer are necessary and sufficient for the gain of resistance, and that the CRISPR-associated or Cas proteins nearby are necessary for the system to work. So in one publication, they transformed the genomic oddity into a bacterial immune system whose broad outlines were already taking shape. This was very impressive, and Doudna and Charpentier labs then went on to build the top of this and demonstrated the minimum Cas9 molecular scissors that we all know and love. And this is where I blank. Okay. Anyway, Professor Barrangou has went on to use the CRISPR system in modern agriculture. He's now based at the North Carolina State University, and he'll tell us in detail about his work. So please join me in welcoming him onto the stage.
All right, welcome everyone, and good morning. Thank you for having me here today. I'm delighted to be here, and I'm quite excited to give you my take and my insights on CRISPR in general and the repurposing of amazing CRISPR molecular machines to revolutionize agriculture. And I've had the privilege to work on CRISPR for 20 years. As a matter of fact, I'm not going to say whether I'm a CRISPR pioneer or not. I think I'm a CRISPR dinosaur, to be honest. My first patent on CRISPR already expired. This means I've had the privilege to see and observe firsthand the amazing journey that CRISPR as a technology has had over the past 20 years in general, over the past 10 years in particular. So on a show of hand, who's actually read a CRISPR paper before? Who's published a CRISPR paper? Okay, and who's consumed a CRISPR product? A drug, a food, no? Anybody invested in a CRISPR company yet? Okay, so let's see how much those answers change in the next 60 minutes, okay? So the business that we're in, and I've been in industry, I was in industry for 9 years at DuPont when I first came across CRISPR and actually commercialized CRISPR. I also worked in academia. I've been an academic for 12 years, and I work in a lab, fundamental lab that does CRISPR discoveries, tool discoveries, Cas discoveries, and deployment applications. But the business that we're in is not just science. The challenge that we have is to do what I call translational science. We have to have great science on the left, and occasionally we know enough about scientific phenomenon, a scientific process, a scientific discovery, a scientific peculiarity, genomic or genetic idiosyncrasy like CRISPR, to turn that scientific knowledge into useful technology. And occasionally that technology is useful enough to be applied. So at JRC, in the Sainsbury lab, you guys use CRISPR as a genome editing technology and apply it in plants. And occasionally those applications are useful enough to be commercialized and to be productized. And the reason I asked my first question is, have you ever consumed a CRISPR product? I can actually tell you right now, you have all consumed CRISPR-enhanced products, unbeknownst to you. And I'm going to illustrate for you in the next hour how those products came about and why they matter so much. And I used to stop at products and commercialization, but in today's world, we understand that acceptance, not just by regulators, matter. And certainly the UK has done a fantastic job as a country to regulate properly or deregulate properly CRISPR engineered products for sure. But we also have to have acceptance by the public, acceptance by the media, acceptance by investors, acceptance by regulators, and acceptance by consumers, patients, and the public. And it's hard to do those things. The activation energy level to go from science to tech, from tech to applications, from applications to commercial products, and for products to societal success and acceptance is hard. Any one of those steps is hard and difficult. But we have to play our part. And this is why I'm excited to be here today, because I'm an academic, right? I'm also a business person. We work with government, right? And of course, we have to work with, and the governments don't always show up, right? As you can tell right here, clearly there's a subliminal message coming through. And we have to work with society. And today, I'm gonna challenge you to have different thoughts and different prisms through which you listen to my talk. Not just as a scientist, but also as a consumer. Not just as a technologist, but maybe as a philosopher, and try to understand, like, where are we with CRISPR? Where have we been and where we're gonna go next, and how you're gonna play a role into that. And I would challenge you to ponder where you want to be as an institute on that spectrum. Where do you not want to be as an institute on that spectrum? And as you think about your careers and your impact in your lives as professionals, where you should be on that spectrum or not, I can tell you surely I'm on the spectrum. All right, so let's start with context. This is CRISPR data. Where are we now? And what has happened in the past 10 years that enable us to foreshadow, forecast, and foretell what's going to happen next. So in a few numbers, CRISPR is explosive. As a matter of fact, Adgene, a repository in Boston, Cambridge, Mass, not Cambridge here, but Cambridge, Mass, ships to not-for-profit organizations CRISPR constructs on a daily basis. If you order your construct by 3 p.m. Eastern time today in the US, you get your construct tomorrow. And remarkably, Adgene has shipped CRISPR constructs 300,000 times in the past decade. Think about this for a minute. 300,000 times a grad student, a PI, or a postdoc have had a request to use from Adgene a CRISPR construct. It's amazing, it's remarkable. Those 200,000 shipments have occurred and been disseminated to 108 different countries. It'd be hard to make a list of 108 different countries, right? And there's parts of the world, I'm not going to point at, that are not perceived as beacons of scientific innovation that use CRISPR all day, every day. That's remarkable, right? It foreshadows where we are, where we're going next. Now it's very competitive, right? You can look at the top 10 list of authors right here. There's over 50,000 people from 5,000 different labs that have co-authored a CRISPR paper, including some of you in the audience. Thank you for your contributions. The US is first, has been first, is not really funded to remain first for a long time at this point in time, but I'll refrain from political commentary today. But you can see China, certainly, has domestically, strategically decided to invest in CRISPR. And their primary focus is your competition in CRISPR and ag. Let's just be honest here, right? And then ironically enough, Germany, France, Holland, and Italy, all in the top 5. But right now in the EU, you're not allowed to commercialize CRISPR. So those governments are investing in technology they can't even use. We'll see by the end of 2025 this changes. Of course, it's very competitive. Of course, it's very dynamic. Of course, it's very international. As a matter of fact, there's 5 different continents in the top 10, right? Americas, Asia, Europe, and even Oceania. So we're talking about a CRISPR world where everything happens. Now, that being said, though, 90% of all that work is occurring in human cells, and 5% of all that work is being deployed in human cell models. That means that 95% of all this is to eradicate disease. That's great for disease research. That's great for medicine. It's great if you're a patient. It's great for therapeutic investors and investments. But it means there's less than 5% focus on everything else. All food, all ag, all forestry, all biotech, all microbes. If you look at this as a tree of life, we're really focusing myopically most of our effort in a limited spectrum. And we need more investments in ag for sure. So stay on the lookout for this and comments of this other paper. And of course, at the bottom right, as you know, there's all kinds of things we can do with CRISPR. It's amazing, it's powerful, CRISPR screens and knockouts and libraries and whatnot. Now let me wear a business hat here. I worked in industry. I'm a serial entrepreneur. I went to business school. I got an MBA. If I look at the same data through that prism, it tells me the same, but also a different story. What does that tell me? Well, it tells me that technology is essentially democratized for scientists. The average grad student at the average institution can do CRISPR experiment the first time, and you expect it to work. 10 years ago, that was another case. The market's global, and this is a co-authorship network of the top 50 most influential CRISPR authors. It tells you that all but two, though it's competitive and challenging and difficult, work together. From my 2 days here, I can tell you guys do a good job working together, but there's a lot of opportunities to work together more with people outside of here. The UK network beckons. The European Union network beckons. I know Brexit notwithstanding, right? And the whole world can enable you to do more CRISPR, better CRISPR, more scalable CRISPR, more affordable CRISPR, and certainly more commercial and translational CRISPR. So collaborations are important. Very opportunistic, I'll talk about that in a few minutes. And then it's very focused. So CRISPR outside of human therapeutics. Where is it happening? Where's the hotbed? Is it here? Is it VIB? Is it Berkeley? Is it China? The question is fair. So how can you be even better at what you do and be more dominant in the ag space? I asked the question. And then, of course, we have to understand there's huge competition, again, in China. And I think once the European Union has the wisdom to deregulate some gene editing technologies, presumably by the end of 2025, we'll have even more competition. All right, and very quickly here, if we mine the data, again, from AdGene, I try to understand where people do what. In blue is therapeutics and in green is ag. Again, a tragedy that overall, this is how much CRISPR ag is occurring in general, in North America, in Northern Europe, in Western Europe, but certainly not in parts of the world where governments are strategically decided to invest their resources for crops of interest to them. And we can't just work on corn and soy. I know you guys love wheat, that's great. Rice is relevant for sure. But there's hundreds of commodities that are in play here, right? Apple cider, case in point. Barley for beer, case in point, right? Tomatoes, but also potatoes, right? There's a lot more to do in CRISPR world than people are doing. And last thing I want to mention here, this is a CRISPR inventions, give or take, and CRISPR users, give or take. So US #1, UK #2, fantastic. US #1, China #2, UK top 5. What's interesting here is look at the ratio of inventors, let's say 10 to 0.6, ratio of users 6 to 1. So when you look at the data, you really have opportunities to understand who's doing what, when, where, and how. And the take-home story here is now is the time, and maybe this is the place, to double down on our investments for CRISPR in agriculture to better the world. Okay, so now that we're here, where are we going next? So the reason I was invited today to give a talk is I've had the great privilege to be part of the CRISPR journey for the best part of the last 20 years. And I have worked on bacteria, I've worked on phages and viruses, I'm working on crops, I'm working on humans, I'm working in forestry, and I'm working with society. And I want you to think through a bigger spectrum of how the work that you have influences other people. You're not just, quote unquote, working on crops per se, because those crops impact livestock, and those crops impact human food. The funding from my lab, as a disclosure, is primarily from industry. Why? Because I try to turn science into technology and apply and commercialize that technology to feed the world and cure the world of diseases. So there's huge opportunities for people like this and people like you in places like here to actually work with companies to engineer the microbiome in order to breed crops, in order to cure human disease, in order to breed the next generation forest. And as a member of multiple national academies, we also have a role to help inform science and regulatory policies. We have to educate not just the next generation of students, but the next generation of politicians, the next generation of patent examiners, the next generation of lawyers, so when they have to decide on CRISPR patents, they are informed. Very important. I'm also a serial entrepreneur, and I've had the great privilege to found 5 different CRISPR startup companies: Locus, phase 2, and Celia Bio in phase 1, in therapeutics in phase 3 clinical trials, and Trico in plant tree field trials. I'll talk about that in a few minutes. And then full disclosure, I'm also an investor and advisor to a number of other companies in the space, most of them for-profit, most of them large, most of them American, most of them in the therapeutic world, but there's a huge impetus to really support and deploy the use of those technologies with big ag, with small ag, and with next generation ag. All right, so let's start with the science. I'm going to walk you through that whole activation process over time and space for CRISPR, focusing on ag, but we have to understand where it comes from, so we'll start with the science. And as you heard, I've been working on clustered regular interspatial palindromic repeats for 20 years. So this is what CRISPR looks like. Anybody seen CRISPR before? No? Oof. Well, at least you see it once in your life. Anybody sees anything? Come on. Yeah, you see it, right? Anybody else see the line? No? I'm gonna help you a little bit. All right, you should see it now, maybe, maybe not. And you should see it now. That's CRISPR. That's my first CRISPR ever. That's what CRISPR looks like in nature. Clustered all together in one location in the genome, regularly interspaced with exquisite periodicity, short about 3 turns of DNA helix, partially palindromic, see the G, T, 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 reverse complement AAC right here, and repeating the same sequences there over and 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 over again. Those CRISPR-Cas idiosyncratic loci occur in 46% of all bacteria on planet Earth. So you think of all the bacteria you have in and on your body right now, right? You actually eat, consume, and breathe CRISPR all day, every day, unbeknownst to you. And as we heard in the introduction, those are idiosyncratic loci that enable bacteria to capture a piece of DNA from invasive genetic elements. We showed that 20 years ago. It's cute, it's interesting, nobody really cares. And the only things you need to know about CRISPR is DNA encoded. You capture a piece of DNA, right? So for the students in the audience, pretend like CRISPR is your phone, right? CRISPR captures with a genetic camera, so to speak, a new spacer from the invasive genetic element to have sequence encoded specific immunity. Once you capture that CRISPR spacer is transcribed into CRISPR RNA that can be synthetically designed as guide RNAs to guide and direct endo and exogenases like Cas9 to do sequence-specific targeting and cleavage. So in nature, this allows bacteria to cut the DNA of invasive viruses called phages, plasmids, mobile genetic elements like transposons, and the like. And there's all kinds of CRISPRs in nature. I'm not going to give a talk on the 2 classes, the 6 main types, and 44 different subtypes. And bottom story is they're all DNA encoded, they're all RNA mediated, they're all nucleic acid targeting. Some target DNA, some target RNA, some are endonucleolytic, some are exonucleolytic, and some are both. So we have different modalities to do different kinds of targeting, different types of outcome in DNA and RNA, and whole books have been written about CRISPR. So I could talk about CRISPR science all day, but to be honest with you, that's not the exciting part. The reason we're here, the reason you came, is because CRISPR as a technology is useful. This is what got Jennifer the Nobel Prize in 2020. You can use CRISPR as Control F in the genome, place the cursor wherever you want at that location. Being in the UK, there was an ad in the Tube yesterday for Sir William Shakespeare's still original theater in London, which I got a chance to go to last time I was in London. And if we were to say, CRISPR is allowing us to rewrite the human genome, and you equate the human genome about 3 billion letters. Those of you who remember phone books in the audience, right? It's about like 10 feet tall of phone books into one human cell. You can Control F to be or not to be, that is the question. Program to CRISPR to place the cursor between that and is, and edit the genome precisely at the site of cleavage. To be or not to be, that's the question. You can add a word, to be or not to be, that is the quintessential question. You can remove something, to be or not to be, the question. Or if you're modern, maybe like to be or not to be, hashtag the question, okay? The point is we have the ability right now to use CRISPR as a molecular scalpel to enable us virtually to edit anything we want, any way we want, in any organism we want. And in genome editing, there's two steps. The first step is cutting. That used to be hard. For those of you who remember genome editing BC, before CRISPR, right? TALENs and ZFNs and meganucleases were feasible, but they were hard, expensive, slow, time-consuming, and you needed a fancy lab with fancy equipment and a lot of time to make it happen. CRISPR has trivialized that part. Again, beautifully in Jennifer's technology, single guide RNA, endonucleotide cleavage, you generate a double-stranded DNA break. The hard part now for genome editing is to understand what DNA repair pathways are in and what kind of edit outcome do we want. Do we want a single base pair change, a random mutation, a small indel, a large deletion, or a large insertion? That is the question. So if there's historians in the room, right, there was genome editing BC, like I said, before CRISPR. But remarkably, 10 years ago, when the technology came out, it took just 1 year for CRISPR to take over incumbent technologies. And it was remarkable. Literally overnight, people realized ZFNs, TALENs, meganucleases are dead. And it went from 400 papers a year to 4,000 papers a year. From a handful of labs are able to do it to hundreds of labs are able to do it. And just 10 years later, remarkably, albeit about $4 billion later, the first ever CRISPR drug was approved as Casgevy by the FDA. So that's remarkable. The speed at which that technology changed the world is remarkable. That's why we're here. And in the past 10 years, those 50,000 authors have been very hard at work publishing, as of this morning, 57,800 papers. Right? It's about 5,000 papers a year. Think about that. Remarkable. And they've developed a very fancy, sophisticated CRISPR toolbox. So I don't have time to go through all the CRISPR toolbox necessarily today, but we can use Cas9 as an endonuclease. We can deactivate the nickase domains RefC and HNH to turn Cas9 into decas. It's not dead Cas9, it's deactivated Cas9. It's still active to some extent, but it's not endonucleolytic. And we can tether to Cas9 other effectors to control and modulate DNA transcription, for instance. Turn things on or off, up or down. And we can also tether dCas9 to effectors like acetylases and methyltransferases that give us the ability to change the epigenome. So for all intents and purposes, we can just change the text, we can change the punctuation. And we have the ability to use CRISPR any way we want, in any organism we want, to control the genome, rewrite the genome, to control the transcriptome, modulate the transcriptome, and to rewrite and recode the epigenetic state of organisms. It's amazing. And by the way, it's not just Cas9, it's Cas3, it's Cas12, Cas13, Cas-TN, CRISPR-associated transposons, base editors, prime editors, ICBs, TNSPs, Fenzor, LSRs, integrases, check, 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 check. And some of my colleagues have made a whole career out of tethering different effectors to deactivated Cas effectors to do not just engineering, but base editing. Think deaminases and Liu, think CAST, Feng Zhang, amongst others, and Sam Sternberg, and then prime editors, David Liu again, pegRNA, reverse transcriptase, and of course, integrases, check, 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 check. We can go into nature and mine for novel effectors that are leaner, smaller, safer, skinnier, easier to port into other organisms and pack into payloads, or at a time of synthetic biology and AI, we can hallucinate new effectors and write them de novo. Think CRISPR meets AI meets synthetic biology and structure-informed scalable screen mutagenesis to give us a toolbox that has everything we need to do. Check. So in the past 10 years, those 55,000 authors have worked on viruses, bacteria, yeast, and model organisms to understand and decipher biology. Great. We've deployed this in organism to make CRISPR or CRISPR-enhanced products. Think like not chicken, but CRISPR chicken, or not just bacon, but CRISPR bacon, so to speak, right? And I can guarantee you, you've all consumed some of those products. I'll go to that in a few minutes. But importantly, the majority of the work, 95%, has focused on humans. To contextualize the pace at which things went, this is from 2014. And butterflies have wing patterns that are the equivalent to the human iris. No two butterflies on planet Earth have the same wing pattern. Control F yellow, take some stem cells out of a butterfly. You can rewrite the code of the DNA of a butterfly that has the exact same wing pattern and has no color yellow in it. We can do the same with the color black, circa 2014. So as of 11 years ago, if you can do that, I make the argument there's no limit to what can be done with CRISPR but our imagination. And as a matter of fact, as the editor-in-chief of the CRISPR Journal, I can prove that to you. You can change the color of the eyes of insects. You can change the color of meat of fish. You can humanize pigs to do genome transplantations in humans and make them stick for a year. You can CRISPR-enhance trees, different cuts of meat, hypoallergenic cats, sustainable agriculture, the editing of the lamb. You can just change one eye color if you want, and we have dosed nearly 1,000 patients with CRISPR gene and cell therapies already, right? On that front, it took us collectively 8 years to dose Victoria Gray. 8 years for patient 1. 1 year to get to 10. 1 more year to get 100. And this year we've dosed 1,000. It's remarkable, right? Not scalable, but remarkable. So CRISPR now is like a 12-year-old prodigy, about this tall, right? And it's won every single prize you can think of. It's an amazing technology, as you can tell here. Programmable, specific, transferable, efficient, precise, affordable, quick, multiplexable, scalable. It's amazing, but it's only 12 years in, right? So think of like, I don't know, phone technology 10 years in, car technology 10 years in, the internet 10 years in, AI 10 years in. It's only 10 years in, right? But 10 years in, it's FDA approved, but we still need to make it better. So it's still a little fat, throws a tantrum here and there. It's not always reliable. We can make it better. We need better guides. We need better Cas. We need orthogonal systems. We need more efficacious, more active, more safe, less toxic, more packageable effectors. And we need to enhance and broaden PAM diversity so we can visually target any precise base in any genome of interest at will. And we're getting there. The hard part, I can tell you right now, are the next 3. Do we really understand DNA repair? I think there's a lot more work that has to be done in non-canonical plants to understand DNA repair in plants. What DNA repair do we want? How much recombination do we want? How much random mutations do we want? And when it comes to large inversions, large deletions, and large insertions, we need to understand that better and coerce that specifically. I'll give examples of this. Delivery is always challenging. Yes, for corn, yes, for soy, yes, for some things like rice and wheat and others. I can choose some of these trees we work on. The limiting factor is tissue culture and understanding delivery. Oh, and by the way, consumers want DNA-free genome editing. So we have to deliver RNP-only modalities of CRISPR for all of the applications that we make. And it all hinges on knowing what to edit. The reason why we've gone so far and so fast in human genetics is because this is all informed by decades and tens of billions of dollars of research on understanding the basis and the rationale for human genetic disease. And in plants, we still don't know a lot of the levers to move the traits we want to change. Also, a lot of those traits, multigenic. So we need multiplex editing. So we need multiplexability across the board to control the genome and the epigenome and the transcriptome. So how far have we gone in applications? Well, now there's a whole world of companies that enable people like us to do genome editing. Buy your CRISPR tools, your CRISPR guides, your CRISPR enzymes, your CRISPR software, your CRISPR kits, your CRISPR plasmids, your CRISPR primers, your CRISPR cell lines. You can even order animal models of human disease and get delivered edited mice in 6 weeks. It's unbelievable. Same thing in biotech. Any bacterium, yeast, or algae used to make food, biomanufacturing, bioprocessing, bioproducts, household care, and bioenergy has been enhanced with CRISPR. So as a matter of fact, next time you do laundry at home, you can thank CRISPR. If you're a student and you can only buy the cheap laundry detergents, They probably have chemicals and scents in them. If you care about your clothes or you can afford the high-end detergents, those have enzymes in them. You need enzymes to take out some stains. You need a cellulase to remove a grass stain, for example. So you can clean the grass stain with soap, but you can't remove all the grass. We work with Novozymes, we work with Genome, we work with DuPont, we work with Novonesis, and pretty much every company that makes every industrial enzyme is using CRISPR to engineer your laundry detergent. That's why you shouldn't eat it, right? You're welcome. Now, the same is happening in food. Any crop, any livestock, any microbe, any tree, any flower, ornamental, grasses, aquaculture, food and seafood, right now is being CRISPR enhanced. They're being bred with CRISPR. Whether they're commercialized or not is the question. But I can tell you in 2011, when I was at DuPont, we used CRISPR to build up phage resistance in Streptomophilus. And Streptomophilus is universally used and sold by DuPont to ferment liquid called milk into yogurt and cheese. If you've had one bite of cheese, one bite of yogurt, one cheeseburger, one pizza, one tray of nachos anywhere in the world since 2011, I can inform you that you've consumed CRISPR-enhanced fermented dairy foods, non-GM. I'll tell you how it works in a minute. And then, of course, if you're a patient, you want gene, cell therapy, antiviral, microbiome therapy, antimicrobial, new transplantation, gene drives, diagnostic, or if you care about your pet, there's modalities you can procure on the market right now that are CRISPR-based. So the reason this is happening so much and so far and so fast is because the science and the technology, as aforementioned, has been repurposed and commercialized and licensed to many companies. I cannot give you a list of all the companies that use CRISPR because it would take all day. I cannot even give you a list of all the companies that are startups that use CRISPR. It would also take all day. These are just startup companies that were incepted based on CRISPR technology. There's over 50. So there's biotech companies, there's publicly traded companies, CRBU, NTLA, CRSP, EDIT, PRME, BAM, NTGI. There's therapeutic companies using CRISPR to alter your genome, your transcriptome, and/or your epigenome. Antimicrobial companies, food and ag companies, check, 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 check. And we're just at the end of the beginning. Now, in my lab, my academic lab, we work at the intersection of CRISPR and microbiology. And we use CRISPR to decipher and manipulate the microbiome. And we use CRISPR to decipher and manipulate the virome. And we mine for novel CRISPR effectors in the dark matter of microbial genomes to develop new CRISPR effectors for genome editing modalities. So in short order, I can tell you we use CRISPR in bacteria to kill viruses. We use CRISPR in bacteria to edit viruses. We use CRISPR in viruses to kill bacteria. We use CRISPR in viruses to edit bacteria. And we use CRISPR in bacteria to edit viruses with other CRISPR to edit other bacteria. I know you may need lunch to catch up, but lunch is next, okay? This is illustrating that the average lab at the average institution can deploy the average CRISPR technologies in creative ways to do anything you can think of. And as a matter of fact, in the past 12 years, we've worked with all those companies to bring CRISPR technologies to commercial reality. So engineer microbiomes in food and ag and human health. Engineering phages to modulate the microbiome, both composition and function, both in food and in ag and in therapeutics. And we've worked with all those companies and lab to bring new CRISPR technologies to reality. CRISPR transposases, Cas9, Cascade, Cas3 type 1, DNA-free modalities, and screening for natural variant for large events. So long story short, I'm going to give you some vignettes of different realms of those applications to contextualize and tell you where we've been, where we are, where we're going next. So you can use CRISPR in bacteria as a vaccination card. Every vaccination event is a genetic barcode, and we can decipher and work with hospitals, the army, or restaurants to understand what bacteria infect what people. You can take a fecal sample, a blood sample, if you have sepsis, an oral swab, sequence the hypervariable CRISPR, compare that to a database, and tell what strain infected what patient, what strain is in what product, and how it's changed in space and time. We can use CRISPR in bacteria to fend off viruses and make yogurt more sustainable. We can go into human pathogens and prevent plasmid carrying antibiotic resistance genes to be uptaken and or disseminated. We can cure antibiotic resistance out of organisms that have them that are undesirable. And of course, if you target the genome of the host, you can do genome editing, you can do translational control, you can screen for large deletions, and you can do targeted killing. So for example, you can use CRISPR in bacteria to target non-essential genes and screen and select for natural instances in which not necessary gene can excise out of the genome. And if you target a genomic island that was provided to an organism through HGT, you can target that, and at least in 1 in a million cases, you can screen for the rare variant in which the island pops out. We've shown in bacteria, you can target islands that are as large as 100 kb and screen for large deletions, transposon-based or homology-based. We also, 10 years ago, showed for the first time you can use multiplexed genome editing to target parts of the microbial genome, especially genomes in bacteria that are not very good at repairing DNA, and use everything you know about CRISPR to do a sequence-specific antibiotic, right? Think of next-generation antibiotics by using the exquisite programmability of CRISPR to target specific pathogenic types and pathogen sequences. So how does that work? So this is a CRISPR locus in yogurt culture called Strathomophilis. There's 32 wild-type strains. This is DuPont Global Culture Collection, strain 7007 Take my word for it, it makes the best yogurt in the world. Unfortunately, that particular commercial strain occasionally gets attacked by phages, and cows are not sterile. This is not breaking news. And milk oftentimes is loaded with phages. So phage 1, phage 2, phage 3, phage 4, those occur in different parts of the world and occasionally yield in losses in fermentation efficiency or fermentation ability whatsoever. We can take that strain, expose it to that phage, let nature run its course, that's why it's now GM, and screen for a spacer acquisition event. And we see that upon becoming resistant to the phage, the sequence acquires a new spacer. You do it again with phage number 2, acquires a new spacer. You do it again with phage number 3, acquires a new spacer. Do it again with phage number 4, acquires a new spacer. And over the course of about 2 weeks, the expert microbiologist at DuPont who knows CRISPR can evolve, using like directed CRISPR evolution, a super stable immune starter culture that is now phage resistant. You can do that in multiple starter cultures using multiple phages across biology and manufacturing sites. So imagine sequencing the virome in cows globally, and/or in milk globally, and/or in milk fermentation facilities globally. And then evolving CRISPR-Cas systems in different starter cultures to blend together super starter cultures that will make cheese forever. I did that for years when I was at DuPont. This commercialized was 2011, and this is how we make more sustainable yogurt and cheese at speed and at scale. This technology can be deployed in any bacterium of interest to do the exact same thing. You can now directly evolve bacteria in any microbiome of interest to become resistant against viruses and control the virome in the microbiome. As a matter of fact, you can engineer it in or let nature run its course. So you can take a strain, expose it to yellow, blue, pink, red, green, and it will have an evolved strain that doesn't occur naturally in nature that I made. And if I see yellow, blue, pink, red, green in that strain, I can tell who made that strain, where it came from, and I can tell it's my strain. And guess what? I did that for years when I was at DuPont in the food industry. I could take a sample out of any of you right now and tell you who made the yogurt or the cheese that you ate last night. For real. I can go to your house, if you make your own yogurt, and sue you for taking my strain out of yogurt that I sold 10 years ago. I will not sue you because I'm nice. But if I see my strain in a competitive product, good luck explaining to a court of law, yellow, blue, pink, red, green was naturally isolated. You can do that in any bacterium of interest, and any microbiome of interest, in any process of interest. And you can tag CRISPR strains. I've been tagging CRISPR strains for decades. I can tell you a lot about CRISPR strains that we have around the world. You can even be facetious and use the DNA code to write into DNA codes. Cysteine, arginine, isoleucine, serine, proline, on and on and on. If I find a CRISPR tag anywhere on planet Earth, I know I made it because it doesn't exist in nature. All right, so we use those kind of approaches across the spectrum of CRISPR modalities in bacteria. So we mine the hell out of bacterial genomes. We develop a lot of tools that are readily available to public. You can look at type 1s, you can look at type 2s, you can look at type 5s, you can look at type 6, you can look at Cas-TNs. And we use those technologies and port them synthetically to do typing, immunity, transcriptional control, genome editing, epigenetic control, imaging, targeted killing, and natural screening. And we use that at the farm level, we use that at the manufacturing level, we use that in the food level, we use that in the human microbiome level. Think next generation probiotics. I'm thinking about lactobacillus, bacterium, bifidobacterium, strep, Akkermansia, Clostridium, check, Lactococcus, check, 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 check. And those are the things we're working on right now. So we're looking at new Cas9s from new effectors to target any synthetic PAM you can think of. We have a paper that just came out last month in PNAS that does that. You don't like GG, you want AT-rich, check. You don't like Cas9, you like Cas12, especially in plants. We have a paper that came out this morning, as a matter of fact, in the CRISPR journal, showing that. And we mine novel Cas12s out of cattle, right? That work great on plant-associated carbohydrate catabolism. They've evolved to work with plants. We work the same in type 1Fs to do large insertions, CRISPR-associated transposons. So CRISPR can co-opt transposases, right, to guide the transposase to do large insertions in nature, type 1F3, type 5K. We can repurpose CRISPR-Cas systems associated with transposases to do large payload integration using CRISPR's programmability with a transposon payload. We can do up to 10 kb and iterate tens of 10 kb to 100 kb. And then, of course, we're tethering some of those CRISPR elements to integrases and recombinases to do single large serine recombinase integration of large payloads in human cells and in bacteria. To tell you how far we've gone, in the context of E. coli, we can use an E. coli host with a Cas9 and code it to target the phage genome and programmably target an area of the phage genome in which we put in a template repair, a repair template that has a deaminase, a base editor. We screen for phages that come out of it that are edited with a deaminase in it, and that deaminase is encoded to target a different host in which you can do deamination of a target locus sequence. So we can use CRISPR in a bacterium to target a phage genome, to put in a different CRISPR to target a different bacterium to do a different edit. In the context of the soil microbiome, we can go into synthetic soil in a mixed microbial community, target a specific organism with that engineered phage to do a specific edit and do a specific phenotype that is detectable. We're working on right now on doing this with CRISPR-associated transposons. We have a paper that will come out next month in PNAS that shows that we can do that with a CRISPR-associated transposon. We have to edit 35% of the genome of phage lambda. So you cannot add 35% of a viral genome like this. You have to take out 35% of the genome first, that's expandable. Replace it with the CRISPR-associated transposons, deliver that to a mixed microbial consortium, and edit up to 20% of the microbiome you are working on. And now taking that out of E. coli to work on variovorics and other soil microbiomes to control soil microbiome phenotypic attributes. Think nitrogen fixation. Think eradicating organisms or pathogens of interest in plants, right? It's microbiome composition and or function. So clearly, any food fermentation, check. Any plant microbiome, check. Ocean microbiome, obviously. Soil microbiome, we have a large project sponsored by the Department of Defense in the US. Of course, livestock microbiome, and microbes in particular, and human gut microbiome modification. So we're talking about infant microbiome for HMOs, we're talking about oral microbiome for oral vaccines, we're talking about vaginal microbiomes for women's health and BV, we're talking about gut microbiome engineering for milk utilization, HMO utilization, fiber utilization, or bile salt hydrolase pool composition modification for C. diff infections and others. Now, the reason you're here, though, and there's a few microbiologists in the room, that's great. But most people care about eukaryotes. After all, we are eukaryotes. So how far along have we gone here? Well, CRISPR medicines beckon. So every week, nearly, there's an article in the New England Journal of Medicine showing a new disease can be targeted and engineered and corrected with CRISPR. So George Daly, dean of the Harvard Medical School, is welcoming officially the era of gene editing in medicine. Sickle cell and thalassemia, Casgevy, the first ever product, FDA approved, TTR in Italia. And there's a large TTR cohort of work happening in London, as a matter of fact. Mariana Fontana is fantastic, Jillian Gilmore, fantastic, working very hard on treating UK patients with TTR amyloidosis for heart disease. Rare diseases, last week, baby KJ in the US, and now the development by the FDA of N-of-1 therapies, enabling pretty much anyone with a genetic disease to have access, theoretically, to a CRISPR therapy. And as a matter of fact, published that a couple of months ago, there's over 108 different clinical trials with CRISPR modalities. You can do ex vivo CRISPR, take cells out of patients, edit them in the lab, and change them, reinfuse them, or you can do that in vivo. All kinds of diseases, sickle cell thalassemia and cancer, TTR, I mentioned, angioedema, hemophilia, infectious disease like HIV, muscle disease like DMD, eye disease, I think there's an eye disease gene editing company in the UK as well, and then even UTI. You can target all kinds of genes with Cas9, with Cas12, base editing, prime editing, LNPs, AAVs, LANT, and even phages. And you can see there's dozens of companies doing that officially, updating every quarter their investors on progress in the clinic. Now, the same modality is being used here and deployed here and many other places in the context of sustainable agriculture. And we need a lot more of crops to be bred using CRISPR modalities. As you know, not just corn, not just soy, not just rye, not just wheat, not just tomatoes and potatoes, but more. There's history, as a matter of fact, right here, of engineering and enhancing health in plants. You know this, I don't need to tell you, you already know that. But we have to have the wisdom to understand what's of specific strategic interest to the UK. How's corn business in the UK? Exactly. Right? Probably more potatoes. I like to see like apples and orchard with cider. Beets, sugar beets. Right? I mean, if we could make CRISPR beer, I'll be the first one to taste it. Maybe today at 5 o'clock, right? Of course, livestock is relevant, but I think plants hold the most potential to build a resilient, sustainable, healthy food supply chain. You know that. As a matter of fact, there's whole reviews written about the deployment of genome editing, base editing, prime editing, CRISPR-A and CRISPR-I, CRISPR-R, and CRISPR-E. The tools are there. Do we have the wisdom to know what to target for traits of interest for sustainable ag? And the answer is yes, but not enough. And I would urge you to not focus so much on CRISPR, because CRISPR is the easy part. I don't want to trivialize. It's the straightforward part. But now is the time, and arguably here is the place, to use CRISPR to engineer carbohydrates. So I mentioned potatoes, for sure. Right? Carbohydrate engineering to enhance fiber composition or dietary enhancement. Dietary fibers, but engineered and made de novo, right? In the context of the human gut microbiome. You guys like wheat. Clearly, So you know everything about protein engineering in wheat, protein composition enhancement. Gluten-free, can be gluten-free at the source, at the base, right? Of course, soy, there's a lot of excitement about soy commercially for sure. As a matter of fact, US is in a very open competition with Brazil to talk about soy. So engaging not just with large US soy companies, but also the Brazilians is something certainly should be of interest strategically to the GIC and Sainsbury Lab. Macronutrient biofortification, I think there's a very strong case to be made there. We all know about golden rice to some extent. But when we talk about consumer benefits and narratives and human health and people, plant, and planet together, now is the time to have the wisdom to know what traits have to be enhanced for better human nutrition. Of course, thanks to Tropic and others, the post-harvest losses, browning makes sense. You know, I think, again, I'm going to argue for better cider, but of course, originally, the non-browning mushroom was there. Same with browning of potatoes. And then, of course, prolonging shelf life. We have the CRISPR tomato. You have the CRISPR tomato, certainly is very much in play. The tools are there. The question is what traits, what genes, what combination, and how much multiplexing we need to do that. But I would urge you not to just focus on the big row crops, but rather crops of interest to the UK for national purposes and to partners that don't know how to do that. I mentioned sugar beets. Certainly sugar cane would be of interest as well. And as a matter of fact, you can see, this is published, again, as of just last year, there's products on the market already, but not just in crops, also in fish. And the FDA, just last month, enabled swine. I think it was April 2025, late April, early May. So we are at the end of the beginning, and we're ushering a new era of breeding by editing in crops and livestock of interest. And I think the key is to pick the right phenotype of commercial value and then infer from the phenotype the genes that have to be targeted. And I can tell you that's what China is primarily investing in right now. So we have to be competitive, we have to be wise. It's not just me saying it, it's the World Economic Forum saying it. And to give you a context of scale, the World Economic Forum is predicting that 100 million farms will have planted CRISPR-edited seeds in the next 4 years. Think about this. We're like 4 cycles away from 2030 for breeding. 100 million farms will have planted by the end of the decade CRISPR-edited seeds in a field trial. I actually think it's an underestimate. How many of these are going to come from here is the question. Right? And which ones? And why and when and how and with whom are you going to commercially partner to bring those modalities and those products to fruition? Sometimes you just need to tell them the target. Sometimes they're going to have to give you the germplasm. Sometimes it's going to be single-plex editing, sometimes multiplex editing, single trait, multi-trait, or trait stacking. You have to have the right ingredients, not just to do the science and the technology, but the application and the commercial deployment to bring value to the collective. But let me finish here with what matters most. And I think by 2030, if we're lucky and expeditious, we're going to dose probably 1 million patients with CRISPR-edited modalities. 1 million patients, option 1. Option 2, we're going to plant 100 million farms with CRISPR crops. We're going to feed over 1 billion people. Option 2. But how about 10 billion people? Are we going to have the wisdom to use and deploy CRISPR in plants called trees to save the whole planet? I asked myself that question in 2019. I gave a talk at a tree breeding conference. There was about 200 people. All 200 tree breeders in the world were here. I mean, they were there, not here. And none of them worked on CRISPR. And I get them to talk on CRISPR, and I behoove them to work on CRISPR. And they seem like curious, but not enthralled. Because they're plant breeders, they're slow. That's not a joke, it's reality. And I realized I was going to have to do it myself because they didn't have the sense of urgency I expected they were going to have. As people who understand everything about forestry, I was perplexed as a non-forester to hear that they're not ready for it. So the last 5 years of my life, I've dedicated one of my 2 shifts at work every day to work on forestry. And plants matter that much when it comes to a global carbon capture. And 2/3 of that is trees. So if I want to start, finish somewhere, it's going to have to be trees. So I partnered with Jack Wang, who inherited the forest breeding program from NC State University, the foremost forest breeding program in the world. And his lab, for several decades, have been working on deciphering tree genetics. And you guys can appreciate how little we know about tree genome. You guys can probably appreciate how hard it is to find somebody who does a tree culture, tree cell culture, tree tissue culture, tree rooting, tree shooting. And I'm not talking about model trees either. I'm talking about real trees for the real world that are commercially relevant, built from scratch. But thankfully, Jack's lab has encyclopedic knowledge about very key pathways that direct lignin synthesis globally in trees. They understand the genomics, the proteomics, the metabolomics of lignin. And lignin linkages and the SG ratio of S-aldehyde to G-aldehyde in forestry matters substantially. And he's work on genotypes, transcriptomics, proteomics, metabolomics, and phenomics to understand how different pathways in trees direct, derive, and influence wood phenotypes, notably lignin content in general, and S2G ratio in particular. And we integrated all that data, worked with world-class AI and ML experts to model all the potential edits, to do genome editing in trees of interest to us, to lower lignin about 20%, double the SG ratio, and keep tree height the same. And there were about 69,000 total possibilities. And of those 69,000 total possibilities, about 350 would lower lignin the way we want, double the SG ratio, and keep the growth phenotype the same. 350 in 70,000, 0.5%. If you don't have AI to do multiplex genome editing, the model says 99.5% of the time, if you just use your genetic insights, you're not going to hit what you want to hit. And the model told us, more often than not, we're going to need 4-plex, 5-plex, and in the majority of the cases, 6-plex genome editing. If you don't have the ability to multiplex genome editing at efficiency, that in 5-plex is about the percentage that you need to get the tree that you want, you'll never get there. So you need tree genetics, you need CRISPR, you need AI, and you need modeling, and then maybe you'll lower lignin enough. Maybe you'll double your CL ratio enough. Maybe you'll double SG ratio for sure, and maybe you get to keep the trees at the height that you want. Fast forward 2 years of work, dedicated hard work in plants called trees, and we're able to grow trees in the greenhouse that have the phenotypes that we want, that maintain the height, and in which we can recode the color of the wood to prove the editing. There's very few things as satisfactory to peel off a tree and expose a bark of a different color to a tree expert that think they all look the same. And we can model lignin content, so we can lower it and yet maintain the same diameter. Of course, you have to work on polyploidy, as you can imagine. And of course, you have to actually measure how much editing you want and dose the editing across genotypes. Now, this is all predicated on understanding the diversity of the genotypes and phenotypes in nature for foresters. Because we can't bring in crazy ratios of SG, crazy amounts of lignin, and not replicate what's within nature for regulatory purposes. So we have to understand, again, what happens in nature in red, and what we can edit in gray. And we pick the right combination to lower lignin content, lower the CL ratio, and double the SG ratio, and maintain the tree height that we want, especially higher growth, higher SG, lower CL, and lower lignin. And you can see lignin content going down as multiplexing goes up, CL ratio going up as genome editing multiplexing goes up, and the edited lines having lignin content that are reasonable, but lower than the wild type, and volumes that is as good, if not higher, than in the wild type. Of course, we have to replicate biological diversity and expand the phenotypic range accordingly. And then you got to do micropulping. We're doing kilos, not tons yet. We even made the first ever CRISPR paper. It's very crisp. And we can do technoeconomic analysis to understand how this is going to impact pulping and processing at the industrial scale. As somebody who worked at DuPont for 10 years, I certainly appreciate that. And we can lower lignin content and lower the demand on the recovery boiler and lower global warming potential by 40%. Same capacity or increased capacity or lower emissions. Same amount of wood, more amount of paper. Lower amount of wood, same amount of paper. Same amount of wood, more paper, less energy, less input, less byproducts, less pollution, fewer enzymes, and more economy. And to scale that up to where we want to do it, we'll be fully at scale by 2048. I could take all day telling you about 2048, how we're going to get there, how long it's going to take. I don't have the time for that. But depending on the trade that you have, 2 to 4 years of greenhouse project, 4 to 8 years, we hope 4 to 6 years of field trial, and then commercial scale up. 100,000 hectares to over 150,000 hectares by 2048. And you can now imagine how this technology, this approach, and those tools can be used overall to manage feedstock for all bioenergy and all bioproducts. Of course, we do grasses, of course, wooded plants, nanomaterials to replace plastics, biofuels, green chemicals, pulp and paper, things like diapers and feminine hygiene products and paper and newspapers and all the Amazon boxes that are there, bioplastic, textiles, advanced structural materials. People want to build whole buildings out of wood right now, okay? And then porous material. We have to be able to do that, as a reminder, DNA free. So we deliver just quote unquote RNPs. So efficient 6-plex, multiplex genome editing in trees with RNPs only requires extreme knowledge about tissue culture, protoplast, and single cell. And of course, regeneration to have shooting and rooting. And we work right now heavily, obviously, on eucalyptus. So in closing here, think through that, right? You have, phase 2 in viruses, phase 1 in the clinic here, full row commercial and field trials, FDA-approved drugs and field trials in the US, going to South America in 2026. You have to use the right tool for the right edit, in the right context, in the right space, at the right dose, and the right time. But with great possibilities come great responsibilities. Now you can wear your ethical hat. Now you can wear your consumer hat, your parent hat, your citizen hat. Right? Are we going too fast or are we going too slow? That's a fair question we should discuss. I can tell you what the patients think. I can tell you what farmers think. I can tell you what ethicists think. Right? How about PR? The last 2 days I've been here, people still reel from the GMO shortcomings of Big Ag. Though it was 25 years ago. In CRISPR years, 25 years is forever, to be honest. How are we going to tell stories, winning narratives, right? And then, of course, how are we going to regulate that? Does it regulate the same or not? I think the UK here is the best template, arguably, globally. The last 2 ministers of Ag have done a fantastic job pushing that internally and be an example for the world. I've opined dozens of times on all those aspects in the CRISPR journal. And the question here is, where do you want to be on the spectrum? Right? As an academic institution, teaching, training people, we want to help inform government policies, patents, and regulators, help collaborate with industry, help work with society, whether it's NC State or JIC and Sainsbury Lab doing it, whether it's industry, whether it's governments, whether it's society. If you look at your own strategic plan, it says it all, right? Your research aims to address global challenges and your knowledge of plants and microbes to answer fundamental questions, check, as well as having significant impact on digital biotechnology, society, and global development, right? You want to research and impact the world. You want to be creative, curious, and answer fundamental questions about science, but translate the knowledge into societal benefits part needs to be focused on. And I think that the 4 strategic programs you have, right? Wheat, check, check, check. Robustness in crops, I think more diversified crops, sustainable food and health, and APH. And I think now is the time, and this is the place to do that. And I invite you all to really ponder, how can you diversify your plant germplasm to get there? How can you take the great science that you have within the Institute, but bring that to industry with startups and licensing and partnerships domestically and globally to bring that to the world? Because the ingredients are here. But I think you have a responsibility to be even more translational to bring that to everyone. So thank you for having me today. We'll take a few questions from the audience.
Thank you, Rodolphe, for a real tour de force of the CRISPR space. Do we have some questions? Students first. Okay.
I'm going to be if you're disappointed if there's no questions. And judgmental. At the bottom right here.
Yeah.
No.
Hi, it was very nice, so thank you. And also you can explain, mention our process. So, we got science and also we come to the stage to accept this everything. But only our problem is not, I think, IP issue, so we can reach more people or industry, right? So with your experience, maybe you can give us advice, like us non-profit organization, how we can handle it.
So I think not-for-profit organizations have a huge role to play, because you're not constrained to making money out of it. It's very freeing. And you have the ability to invest in crops that are orphan crops, that are needed, or traits that don't have tangible commercial value but have huge societal value. So I don't want to oversimplify that, but a lot of industry cares about yield and disease resistance. And they care about yield and disease resistance in major areas of the world. We need to care more about nutritional composition and disease resistance in other parts of the world where the build programs are not sophisticated. So we have to work with local breeders. I think actually India is having a tough week, but India is a very important part. And that's where the IGI, for instance, is helping Indian rice breeders to breed specifically locally for local diseases to prevent crop loss for the Indian market.
Actually, we are working with, again, Simit and other companies, also some commercial companies in UK and also other institutes about target. So we pass all the stage, we target the real with variety. So only stuff we produce the prebiotic lines, but only stuff is the IP issue and also the regulations EU. So that's why breeders doesn't want to use our prebiotic lines. And also we can use CRISPR for research purpose, but breeders, and if you start to work with stuff, they don't want to use it because they have to pay maybe.
Yeah, they have to pay for IP for sure. So let's start with regulation first and IP second. So regulations in the UK are the best in the world. So you're good here. And I would predict with some level of confidence that by the end of 2025, the trilogue for NBTs will wrap up in the EU. So the meeting in Denmark in September will be very important for that. But let's presume or assume that by the end of the year, the regulations in the EU will have gotten it right, will be implementable and turn into law post-trilogue, and that then the rest of the world is going to be good there. So regulations, I think we're almost there. The issue of the IP is a fair issue. When it comes to orphan crops and small-scale commercial operations, you can negotiate very agreeable, not reasonable, but agreeable terms with license providers, because they want to do what's right and they want the narratives as well. That's a very important aspect of the commercial path for sure. If it comes to big crops, you're going to have to negotiate into it and understand that there's going to be a price to pay for access to the tech for sure. I like Cas12 more than Cas9 implants for all the things we need to know. So the Cas9 IP is maybe mostly irrelevant here. And there's different pools of Cas12 IP you can get from different providers. I can't articulate, but we can surely discuss that behind closed doors. But you have to remind people that though the CRISPR foundational IP and license is useful for the breeding process, it's only solving part of the problems. And some of those high licensing fees people expect and milestones they expect are not commensurate with the relative value provided by CRISPR. When you think about tissue culture, when you think about knowing the traits, when you think about being able to implement it. And also if you need to have a pool of licensing fees you're willing to pay, they can only claim a piece of that pool, because if you have to stack the strain from somewhere else as well. My experience of that as an expert negotiating entrepreneur is people understand those arguments and you can make deals. But you have to be aggressive and you have to be transparent and you have to be straightforward and you have to use leverage from multiple providers to get to where you need to be, but it's doable.
Okay, we're gonna end it here because it's getting late, but if you have any burning questions, you can email us and we'll forward them to Rodolphe. And there's also a couple of questions in the chat room, but they'll also get forwarded and then we can send the responses back. And now we have the student lunch and we have some free spaces on the student lunch. So if anyone is hungry intellectually as well as physically, they can join us. Thank you very much.